Our next speaker is Michael Kohanski from Penn. He's an associate professor in otorhinolaryngology. Take it away.
Thank you. It's a pleasure to be here today. Let's see.
That's okay.
So I became interested and was introduced to this field when I started as a fellow in Noam Cohen's lab, and I think it's really exciting to be here to be able to honor Dr. Margolskee. I think that this work really wouldn't have been possible without the strong collaboration between our group and the whole Monell team. And so I was certainly intrigued by all the functions of Tuft cells, which is kind of where my head went initially when I kind of first got into this. But Noam kind of tasked me with this question, which I'll get into in a second, is what's going on in polyps, which is a subtype of chronic rhinosinusitis that's characterized by type 2 inflammation. And are there more of these cells, fewer of these cells? And I'll kind of get into kind of why we're interested in that and how that's kind of led me into how do we make more or fewer of these cells. So I'll start off just with a super duper brief background on Tuft cells in the context of CRS with nasal polyps. There's a lot of work that's been done, particularly in the last decade, on some of these inflammatory models, and then really get into what we're doing in human nasal Tuft cell differentiation in CRS with nasal polyps. And I will say that all the work that we're doing, all the work I'm showing today, is using primary patient samples from the sinonasal epithelium. So this is all human-driven work in the lab. So just super briefly, I missed Noam's talk, but this is probably the same slide. But CRS, super common disease, pretty big impact on quality of life. Prevalence is somewhere probably between 10, 15, 16%. Costs a lot of money to the healthcare system. And, in our kind of very simple way of thinking about it from a phenotype perspective, there's the with polyp group, which is characterized by these oftentimes bulky inflammatory polyps that can cause various levels of symptoms in patients, or the non-polyp group, which we also oftentimes think about associated with bacterial infection. And so CRS with nasal polyps, as I mentioned, has a pretty strong type 2 inflammatory relationship. It accounts for about 20% of all cases of CRS. And about 40 to 67% of patients who have polyps also have comorbid asthma, another chronic type 2 inflammatory respiratory disease. And those patients who have polyps also tend to have more severe asthma at baseline as well. And so in the context of Tuft cells, one of the things that we're interested in is, can we regulate or detect the disease status through modulation or detection of these sinonasal Tuft cells? And the reason we're interested in this has to do in part with these three papers that came out in 2016. And Dr. Margolskee, I know his group was involved with the paper from Michael Howitt, where really showed that these Tuft cells are sensors of mucosal type 2 immunity. And really, this is this mouse gut model for hookworm infection, which is a type 2 inflammatory model, where the Tuft cell is somehow able to sense the hookworm, and it releases IL-25, which is thought to be constitutive in the mouse gut, recruits ILC2s, those secrete IL-13. You get more Tuft cells, a robust inflammatory response. You get more goblet cells that are made with IL-13, and then the infection is cleared and everything goes back to normal. And so the question was, we have this type 2 inflammatory disease that we're interested in with nasal polyps, and are there more Tuft cells in this disease state that would reflect what was shown in the mouse gut? And we were able to show this, that indeed there are more of these Tuft cells that we can see in nasal polyps compared to non-polyp epithelium from patients who do not have chronic rhinosinusitis. So since then, there's been other papers kind of along the same line. So Myakotis had a paper now from a few years ago, which also showed that there is this increase in Tuft cells in nasal polyp compared to non-polyp epithelium. And one of the interesting findings from this paper is this idea that the Tuft cells may be a little different in the human nose, in terms of that they're maybe activated or mature, and that something was happening with IL-13 that was changing the function of the Tuft cells. And this was really kind of put forward in the context of prostaglandin mediator synthesis and release. And so the way I kind of think about how Tuft cells are working in regards to the epithelium is that you have this series of inputs that are moving through your epithelial layer, and the Tuft cell is really a signal amplifier in a lot of these situations, and it helps to modulate some of these different outputs. So, some of those super basic questions I was interested in is, can we better identify and work with these human nasal Tuft cells in vitro? And if we're able to manipulate them in culture, we can then do some more interesting experiments. And particularly this kind of led to this idea is, what are the mechanisms that are driving nasal Tuft cell differentiation? And with the idea is, can we at some point come to a conclusion of, are these cells bystanders, or are they actually contributing to pathogenesis in CRS with nasal polyps? So the first thing is we took a step back, and one of the things that we've learned is that not all markers that work in the mouse work in the human. And we went into our single cell data set that we had from nasal polyp patients. And, we had a list of some of these genes here that came up. And one of the ones that was super high up as a good target to go after was this gene BMX. And we looked at other single cell data that was available as well, and we see BMX pop up there as well. It's enriched in these polyp Tuft cells compared to non-polyp. And there's quite a bit that's actually known about BMX. It's this bone marrow tyrosine kinase gene in chromosome X. It has some, it's best studied more in, around blood cancers and things like that. But we don't actually know anything about what it does in a Tuft cell. And we still don't know what the answer is to that either. So what we did at this point is we went into our tissue culture model that you've heard about a little bit in some of the other talks as well. So the basics of this is we take our sample from a patient, we can generate a submerged culture of what's mostly basal cells, grow them for about a week till they form a monolayer. Then we take the fluid off the apical surface, transition it to a differentiation media for about 3 weeks, and we can get this differentiated air-liquid interface culture, which has ciliated cells, goblet cells, Tuft cells, other rare epithelial cell types in it as well. And so one of the things that we wanted to know is, do we see BMX in Tuft cells? And so what we did is we looked at, by immunostaining in some of these cultures, looking at Pu2F3, the main transcription factor for Tuft cells, and we see very nice co-localization of Pu2F3, which you can see here in magenta, with BMX in green. And we did this with several other markers as well. We can see good co-localization of BMX with TRPM5. And importantly, we did not see any co-localization with FOXI1. So just to kind of bring in ionocytes really briefly, there's some data that there may be this common Tuft ionocyte progenitor cell. And so we're not seeing BMX co-localizing with FOXI1, suggesting that it's probably a more robust marker of either this mature or at least some variant of the Tuft cell. And so another kind of basic question is, okay, we have this whole culture model. Where are the Tuft cells? When are they showing up? How do we kind of pick the best time to kind of trigger them to amplify? And I'm going to summarize a lot of negative data by saying that you can't get Tuft cells in submerged cultures in the conditions that we've looked at. We just have not been successful in that condition. And so we really honed in on this air-liquid interface culture model and the transition to air, looked at various time points with different techniques. And really what we found is that over time, you start to see increased expression, increased presence of these various Tuft cell markers. And this fits with what's been published in some of the bronchial epithelial cell lines as well, where these different markers appear over time as the culture matures. And so we know that IL-13 is a really good driver of Tuft cell amplification, and really using that as a basis for driving Tuft cell hyperplasia in this in vitro model. And so we wanted to then see, does timing or duration of treatment matter in terms of generating Tuft cells? And so we came up with these 3 different models. We had what we called a chronic IL-13 model, which is basically marinate the culture in IL-13 through the entire growth and differentiation process. And then we had a condition that we called post-diff, which was we took a fully mature ALI culture and then hit it with IL-13 for a couple of days to see if there's some progenitor or cell that's still there, can it become a Tuft cell? And then we also had this pre-diff model, which is when the cells were transitioning to air, you have basal cells that are differentiating. If you hit at that point, do you get more Tuft cells. And so we did this by immunostaining, looking at the Pu2F3 positive and then this double positive subpopulation. You can see those here on the x-axis. And on the y-axis is the number of cells per high power field. And what I didn't mention is that one of the ways we're trying to overcome patient-to-patient variability from sample to samples, we're actually pooling cultures here from our patients who have the same disease phenotypes. This is all done in our CRS with nasal polyp phenotype. So each dot is basically, it's the average from several replicates of a series of pooled cultures. So we're looking at, numbers from somewhere between 18 to 24 plus patients in this data set. And the first thing right off the bat you can see is that IL-13 increases Tuft cell abundance, and it doesn't seem to matter when you do the treatment. And so what's also interesting is that this repetitive IL-13 treatment over time, you don't continue to get increased Tuft cell development over time. So there's not the self-replicating pool. There's some fixed pool that's present in our culture condition that has the ability to turn into a Tuft cell. And so, the next question we asked was, okay, so this is all done in polyp. We put IL-13 on. Is this phenotype specific to CRS with nasal polyps? And, there's some data from what we've done, from others have done, suggesting that, again, there's this Tuft cell enrichment in nasal polyps. And there is also this some evidence that of an idea of inflammatory memory with basal cells in nasal polyps, that they're able to essentially remember the inflammation. So remember, we're taking the cells out of the patient, and they're going into this predefined culture media. So how do they remember basically where they came from? And so what we basically did this experiment. Here's our polyp data. This is using the model where we took the fully differentiated cultures and treated them for 3 days with IL-13 and then measured the Tuft cell numbers. And we, again, see this increase in Tuft cells. And when we did this in our healthy ethmoid control, so the ethmoid sinus is kind of what we think of as kind of ground zero for where sinusitis starts from. And these are patients who are undergoing surgery for tumors or other reasons. We do not see an increase in Tuft cell expansion in this population. And we asked the same thing in our, what we think of as more of our type 1 inflammatory group, this CRS without nasal polyp, again, the same thing. And so this suggested to us that the Tuft cell phenotype with this increased abundance appears to be specific to the nasal polyp phenotype. But what I will mention also is that this is all done in this standardized media, which we get from this company. It's called Pneumocult Media. I have kind of no stake in anything that they do, besides the fact that it's proprietary and you can't get them to tell you what's in the media. But we do know that the initial media that is probably designed off of was really designed, and this whole system is based off of promoting ciliation and looking at transport. And we've kind of co-opted this model over the last 2+ decades to look at other functions, other cell types, innate immune functions. And so the question was, could we tune our media, tune our growth conditions to optimize Tuft cell increases with IL-13? And again, summarizing a lot of negative data before I get into some positive data, we ended up settling on this idea of serum starvation, which is really used a lot more in fibroblast cultures where you can put the cells into a quiescent or a stress condition. And we also removed retinoic acid, which is an important differentiation factor for driving ciliation. And we basically replicated this model where we differentiated our cultures in the normal media, flipped them to this reduced retinoic acid-free media with IL-13, and then measured Tuft cell abundance. And what we see here is when we flip the condition here, we are able to essentially unlock this phenotype across independent of disease states. So you get an increase in Tuft cell hyperplasia. It doesn't matter what the disease state that you start with. And what we found, and you can see that just in the red is with IL-13, blue is without. And what we found is that when you come back in and add retinoic acid into this media, at least in the CRS phenotype, you're able to then drop down, at least to some degree, the amount of Tuft cell expansion that you get, suggesting that retinoic acid is playing a role as a brake on Tuft cell differentiation. And so with this in mind, we're trying to dive into what are some of the mechanisms, from what's known about IL-13 and retinoic acid. IL-13 classically signals through STAT6, and trying to understand what are some of the transcription factors and processes driving this. And just, super quickly, we are relatively confident that this is a STAT6 independent process. So just from some Western blot data looking at phosphorylated STAT6 from healthy controls compared to polyp, you can see that with IL-13, you get nice STAT6 phosphorylation. And when you add retinoic acid, and it doesn't seem to make a difference there. And again, with ALOX15, which is known to be regulated by STAT6, goes up with IL-13, and retinoic acid does not seem to affect it. But we do see an effect when we look at Pu2F3 expression. So trying to dive more into that. So just to wrap it up, BMX we think is a marker of these mature or a subtype of these nasal tuft cells. Our in vitro model suggests that there are intrinsic differences in tuft cell differentiation between polyp and non-polyp phenotypes, and that with this reduced retinoic acid-free media, we can kind of unlock this differentiation across disease phenotypes. And just trying to dig in and see if we can identify some novel therapeutic targets at the end of the day that can reduce type 2 inflammation and polyp recurrence in this disease state, because we think that these cells might be an important driver. And with that, just like to thank Deepa and Tiffany, who've really kind of done the brunt of the work in the lab, to Noam, Dabrowski, Andy, Jim, Nithin, John, Jen, and Rob for all their help, and to Danny and Kailu as well for their help with all the analysis of the single cell data. Thanks, and happy to take any questions.
Thank you. Given that the BMX sends a fixed chromosome, and that in the rat biliary tract and in the mouse urethra, there's really a sex difference in the development of these stomach cells. So they come even 1 week after in female than they come in male. Have you seen any sex differences in your cultures that will separate, let's say, male and female bonus?
That's a great question. We have not yet separated, at least in our cultures, based on sex. It's something that we should be doing, but sometimes we're limited by what we have coming through the door. I have been trying to get up a knockout mouse for this, which does exist. And if there's a paper from 2000 or 2001, there's a Laxi BMX fusion mouse, and if they have a picture of the tongue, and like it looks like the taste buds are just lighting up with the beta gal assay and that. So it's probably there. It's probably doing something. We don't know if it's playing a role in function or differentiation. That's a good idea.
The mice get nasal polyp?
No. Yeah. So that's the secret of this, is that there's not a really good model for nasal polyps in general. And a lot of the models in the mouse, I think there's questions, is it really a chronic model or an acute model that you just leave for a while?
So you had several great markers for the mature tuft cells, but you went with the one that no one's ever heard of. So what was the rationale there?
We tried some. We didn't have antibodies available. Some we tried. We couldn't get the antibodies to work. And we started with one and we just kind of ran with it. And it was compatible with Pu2F. And so that was a big driver for that, for sure. I have thought about that in retrospect.
You called this differentiation. Can you recognize an immature tuft cell that then differentiates into what you're calling a differentiated tuft cell?
Maybe. So there are several papers on that. And Dr. Cleavers didn't talk about it, but he has a paper in the gut where he has a marker that they think can do this in their organoid model. And there's some evidence in the airway as well. And we've been able to sort on that marker. And
what's the marker?
c-KIT. So it's not really expressed super highly in the airway and epithelium in general, but it seems to be there. We were excited to try it. We've tried it a bunch of times. We think it's probably working because when we put IL-13 on our cultures, we lose it. So, but we'll see.